Long ago, people believed that the world we see with our eyes was all there is. To them, a drop of water was nothing more than a clear liquid, and a patch of mold was just a stain on bread. But when the microscope was invented, everything changed. Suddenly, hidden worlds were revealed. A simple glance through the lens showed that water was teeming with tiny moving creatures, and mold was made of delicate threads and spores. The microscope became our new set of eyes, allowing us to see what the human eye alone could not. Through it, we discovered cells, the building blocks of all living things. We realized that diseases were not caused by curses or bad air, but by invisible bacteria and viruses. The microscope gave us the power to peer into the structure of plants, animals, and even our own bodies. What we see today, the cells dividing, the intricate shapes of microorganisms, the beauty of crystals and tissues, is not just because of our natural vision, but because of the microscope. It is a window that expands human sight beyond its limits, reminding us that the world is always more than meets the eye. With that, come join us to unravel the world of phase contrast microscopy. So what is phase contrast microscopy? Phase contrast microscopy is based on the principle that the cells differ in reflective index from their surroundings. Reflective index is the ability of the material to alter the speed of light. Light passing through a cell thus differs in phase from light passing through the surrounding liquid. This subtle difference is amplified by a device in the objective lens of the phase contrast microscope called the phase ring, resulting in a dark image on a light background, just like the image shown in the video. So now, let's describe the differences between magnification and resolution. So, let me ask you, what is magnification? Magnification describes the capacity of a microscope to enlarge an image. It is also important to note that all microscopes employ lenses that provide magnification. However, magnification is not the limiting factor in our ability to see small objects. As for the magnification of a phase contrast microscope, the magnification range of a PC microscope is usually of 4 times, 10 times, 20 times, and 40 times. Higher objectives are also available. Examples include 100 times magnification in oil. It also uses corresponding condenser annuli that is matched to objectives. Higher magnification means smaller faceplate, width or diameter, and larger condenser annulus. Now, we tackle resolution. So what is resolution? Resolution is the ability to distinguish two adjacent objects as distinct and separate. It is actually the resolution that governs our ability to see the very small things we observe under a microscope. Additionally, resolution is limited by the diffraction of light, usually about 200 nanometers under optimal conditions. Therefore, the phase contrast microscope operates close to the maximum resolving power of light microscopy.
The regular light microscope that uses stains and dyes to view specimens, the phase contrast microscope uses differences in the refractive index of cellular structures to generate contrast using specialized optical components. Now, what makes this microscope unique are three key parts that work together to produce the phase contrast effect. These are the annular ring, the phase plate, and the phase telescope. Each of these components has its own important role. The annular ring shapes the light, the phase plate modifies the phase of that light, and the phase telescope ensures that everything is properly aligned. In the next sections, we'll look closely at each of these parts one by one to understand how they function and why they are essential for phase contrast microscopy. The first key part of the phase contrast microscope is the annular ring, which is found in the condenser. It is basically an opaque flat plate with a transparent ring in the center. What it does is very important. It allows only a ring or circle of light to pass through, instead of letting light spread evenly like in a normal light microscope. This ring of light is then projected onto the specimen. But its role doesn't stop there. It also shapes the light into a hollow cone. You can think of it like a spotlight that doesn't shine in the middle but only around in the edges. This special light pattern is what makes phase contrast possible, because it is designed to match with the phase plate inside the objective lens. Without the annular ring, the phase contrast microscope would simply act like a normal bright field microscope, and we wouldn't get the extra contrast needed to see transparent structures. That's why this small but critical component is considered the starting point of the whole phase contrast process. Now that we've seen how the annular ring shapes the light into a hollow cone, the next step happens inside the objective lens with the help of the phase plate. It is mounted near the rear focal plane of the objective. It looks like a transparent plate, but it has a special ring filled with materials that can either advance or retard the phase of light waves. What this means is that the direct light, which passes through the annular ring, is slightly shifted in phase compared to the scattered light coming from the specimen. By selectively altering both the phase and amplitude of these light waves, the phase plate creates differences in brightness and contrast that our eyes can detect. This is the key to turning normally invincible, transparent structures into visible details. So, if the annular ring provides the unique pattern of light, the phase plate is what actually transforms that light into a uniform contrast that we can observe. However, for the phase contrast effect to work properly, the annular ring and the phase plate must be perfectly aligned. And this is where the third unique part comes in, the phase telescope. The phase telescope is a special eyepiece that temporarily replaces the normal eyepiece. When we look through it, it allows us to see both the annular ring in the condenser and the phase plate inside the objective lens at the same time. This makes it possible to check if the two are aligned. If they are misaligned, the phase contrast image will be poor or uneven. By using the phase telescope, we can make adjustments until the rings are perfectly matched, ensuring that the microscope produces the proper phase contrast effect. So, while the annular ring and phase plate create the effect, the phase telescope plays the crucial role of making sure everything is set up correctly.
As for the light source of the phase contrast microscope, first we discuss phase object. So it is the unstayed specimens that do not absorb light. This is because they slightly change the phase of light that is diffracted by them. The light is usually phase shifted by about 1/4 wavelength compared to the background light. Our eyes are unable to detect this light phase differences as they can only detect variations in the frequency and intensity of light. Phase contrast enables high contrast images to be produced by further increasing the differences of the light phase. It is this characteristic that enables background light to be separated from specimen diffracted light. The difference of the light phase is increased by slowing down or advancing the background light by a 1/4 wavelength, with the phase length just before the image plane. When the light is focused on the image plane, the diffracted and background light cause destructive or constructive interference which decreases or increases the brightness of the areas that contain the sample in comparison to the background light. So the diagram on the left shows us the path of light in a phase contrast microscope. So to further discuss, the light from a tungsten halogen lamp goes through the condenser annulus in the soundstage condenser before it reaches the specimen. So this allows the specimen to be illuminated by parallel light that has been defocused. Some of the light that passes through the specimen will not be diffracted. These light waves form a bright image on the rear aperture of the objective. The light waves that are diffracted by the specimen pass the diffracted plane and focus on the image plane only. This allows the background light and the diffracted light to be separated. The phase plate then changes the background light speed by 1/4 wavelength, as discussed earlier. When the light is focused on the image plane, the diffracted and background light will cause destructive or constructive interference, which changes the brightness of the areas that contain the sample in comparison to the background light. Often, the background is also dimmed by 60 to 90% by a gray filter ring.
This diagram shows the phase contrast microscope. Aside from the eyepiece, objective, and base, it has collimating screws for light alignment, a moving table for the specimen, and a reflector. These parts convert light phase shifts into contrast, making transparent specimens visible. Here are real images of the microscope invented by Fritz Zernike, who won the 1953 Nobel Prize. It looks like a regular microscope, but its optics allow clear viewing of live, unstained cells and microorganisms.
Now that we've seen how phase contrast microscopes are built and how light travels through the system, let's move on to their applications and what we actually see under these microscopes. Phase contrast microscopy is most valuable because it allows us to examine transparent living specimens in their natural state without killing, fixing, or staining them beforehand. This is especially important in biological and medical research, particularly in cytology and histology, where we can directly observe living cells, tissues, and microorganisms. Internal components such as cell membranes, nuclei, mitochondria, mitotic apparatus, cytoplasmic granules all become clearly visible using this microscopy technique. Beyond that, phase contrast is also widely used in hematology, virology, bacteriology, parasitology, paleontology, and even marine biology. We can see here that when we actually view specimens such as bacteria, we will notice that the normally transparent cells stand out against the background because of the differences in refractive index. These differences are converted into differences in brightness. This will reveal fine cellular detail, though the images often appear soft and slightly blurry because we are already at the limits of optical resolution. Another hallmark of phase contrast microscopy is the bright halo that appears around cells, which you can see in this slide. Next slide, please. Halos occur because the circular phase retarding and neutral density ring in the objective faceplate transmits not only the undiffracted surround waves but also a small degree of diffracted light from the specimen. The effect is amplified because the width of the Sirwet order surround wavefront is smaller than the faceplate ring itself, typically by about 25 to 40%, which is a design compromise necessary for the optics to function. So in summary, phase contrast microscopy opens a window into the living cell, letting us record dynamic biological processes with striking clarity, while also reminding us of the optical trade-offs that come with this powerful technique.